If you were to unravel all the DNA inside your body right now, like untangle every microscopic thread from every single cell and just lay it all end to end, it would stretch from here to the sun and back.
Right.
Not just once either. It would make that round trip roughly 600 times.
Yeah, it's, you are essentially carrying over 60 billion miles of biological code, and it's all folded up so tightly inside you that it is completely invisible to the naked eye.
Which is just wild to think about. And yet, despite that staggering, mind-bending scale, if a single invisible chemical letter in that vast distance is out of place,
Just one.
right? If just one microscopic puzzle piece doesn't snap together the way it should, the entire architecture of a human body can begin to break down. So today on this deep dive, we were opening up the ultimate biological filing cabinet. We're going to look at the master blueprints that built you, me, and literally every living thing on this planet.
Yeah, we usually just marvel at the finished building, right? Like we look at a skyscraper and admire the glass and the steel.
Yeah, exactly.
We don't really spend much time thinking about the actual filing cabinet in the architect's office, but the blueprint dictates the building. So today, we are exploring that journey from the hidden, abstract blueprint to the physical glass and steel of the human body.
And before we take another step, I want to make a promise to you, the listener. If you are tuning into this deep dive, whether you're walking the dog, commuting in traffic, or just, staring at the ceiling on your couch, you can completely relax.
Oh, absolutely.
We are tossing the heavy biology textbooks right out the window. You do not need any prior biology knowledge to follow along with us today. We are going to keep this completely simple, straightforward, and entirely free of thick, confusing jargon.
No pop quizzes at the end.
Right. We aren't going to make you memorize intimidating chemical names. We just want to have a conversation about how the fundamental machinery of life physically works.
Because honestly, biology can easily get bogged down in terminology. But when you strip all of that away, the underlying mechanisms are incredibly elegant.
They really are.
They're just physical shapes fitting together. And everything we're going to unpack today falls under one overarching universal rule. It is the absolute foundation of biology, and it's often referred to as the central dogma.
The central dogma, that sounds like the title of a sci-fi thriller.
It does, right. It carries a lot of weight. The term was actually coined back in the 1950s by Francis Crick, who was one of the scientists who helped deduce the physical structure of DNA.
Oh, okay.
But beneath that heavy, kind of intimidating name, the central dogma is just a simple three-step sequence. It just describes the one-way flow of information in every biological system.
And what's the rule?
The rule is this: DNA makes RNA, and RNA makes proteins.
DNA makes RNA, and RNA makes proteins. That is the entire secret to life.
That is the core engine of life. Information flows in that specific direction. So the DNA holds your master instructions. The RNA acts as a temporary messenger carrying a copy of those instructions.
Got it.
And then the proteins are the final physical products. They're the microscopic machines that make up your muscles, digest your food, fire the neurons in your brain, and literally build everything functioning inside you.
Okay, let's unpack the sequence, starting at the very beginning. We have our three main characters, right? DNA, RNA, and proteins.
Right.
But before we can understand how the cellular construction crew actually builds a bicep or, a piece of hair, we have to look at where the instructions to do that are kept. Let's start with character number one. What, on a purely physical level, is DNA?
Well, DNA stands for deoxyribonucleic acid. And in human cells, and actually in the cells of plants, animals, and fungi too, this genetic hard drive is stored safely inside a highly protected central vault.
Right.
And we call that vault the nucleus.
So this is where we usually see that famous double helix image, right? The twisted ladder.
Yes, that twisted ladder shape is key to everything. And the discovery of that shape is a famous story in its own right. Yeah, back in the early 1950s, a brilliant chemist named Rosalind Franklin used this technique called X-ray crystallography to take a picture of DNA. She basically bounced X-rays off the molecule and captured the scatter pattern.
Taking a microscopic photograph.
Exactly. And that image, which is known as photograph 51, revealed this undeniable X shape. And mathematically, that was the signature of a helix. Watson and Crick later used that data to realize the molecule was essentially a ladder that had been twisted tightly into a spiral.
And what is that twisted ladder actually made of? Physically.
Well, the long sides of the ladder, like the handrails you'd hold onto if you were climbing it, those are made of alternating sugar and phosphate molecules. They just form a really strong structural backbone.
Okay.
But the real magic is in the rungs of the ladder. Those rungs are made of chemical letters, which we call bases. And there are only 4 of these letters in the entire DNA alphabet.
Wait, only 4?
Just 4. Adenine, thymine, cytosine, and guanine. But for the sake of keeping it simple today, we're just going to call them A, T, C, and G.
Just 4 letters. That always stops me in my tracks, because you think about like a mushroom in the forest, a blue whale in the ocean, and me sitting here in this chair. We are all completely different, but our instruction manuals were written using the exact same 4 letters.
It's pretty amazing, isn't it? The staggering complexity of life doesn't come from a massive alphabet. It comes from the nearly infinite ways you can arrange those 4 letters in a sequence.
Right, the combinations.
Exactly. But the most crucial mechanical feature of DNA is how those letters actually pair up to form the rungs of the ladder. They don't just attach randomly.
Okay, so how do they attach?
Adenine physically only fits together with guanine. So A always pairs with T.
A to T.
Right. And cytosine physically only fits together with guanine. So C always pairs with G.
They snap together like highly specific puzzle pieces, A to T, C to G.
Yeah, because the physical shapes and the magnetic charges of those molecules mean they can literally only lock in with their specific partner. And this pairing rule is the foundation of how DNA copies itself.
How so?
Well, if you pull the 2 halves of the ladder apart, you see an A on one side, you automatically know a T has to go on the other.
Oh, I see. So the other side is perfectly predictable. Okay, I want to introduce an analogy here to help ground all of this, something we can carry through our entire journey today.
I love analogies. Go for it.
Let's imagine the inside of a cell as a bustling walled city. And in the very center of the city is a massive, heavily fortified library. That's the nucleus.
Perfect.
Inside this library, sitting on a pedestal in the most restricted section, is an ancient, priceless master reference book. That is the DNA. It holds the ultimate instructions to build and repair everything in the city.
Right.
But because this master book is so incredibly valuable, there is a strict rule. You are never, under any circumstances, allowed to check this book out. It cannot leave the safe confines of the library.
That captures the dynamic perfectly. The cell goes to immense lengths to protect the DNA. Think about it. If the DNA in the nucleus gets damaged or mutated, every single copy made from it going forward will be fundamentally flawed.
The original has to stay perfect.
Exactly. The original master blueprint has to remain pristine.
So it's locked away. But here is the question that always kind of bothered me about this setup. Why is DNA so uniquely suited to sit in a vault for an entire human lifetime without breaking down?
Yeah.
What makes it chemically durable enough to be the ultimate master copy?
That is a great question. And the secret to its incredible stability is actually hiding right there in its name. Let's look at the word. Deoxyribonucleic acid. The prefix deoxy literally means it is missing an oxygen atom.
Wait, missing an oxygen? We generally think of oxygen as the thing keeping us alive.
Sure.
How is lacking oxygen a good thing for a biological molecule?
Right. It sounds counterintuitive. Oxygen is absolutely essential for our lungs, yes.
Yeah.
But purely from a chemical perspective, oxygen is a highly reactive, greedy element.
Greedy how?
It constantly wants to pull electrons away from other atoms. It loves to interact, form new bonds, break existing ones. You can think of oxygen acting on a molecular level a bit like rust on an old car. Rust is literally oxidation, right? It's oxygen ripping metal apart over time.
So oxygen is basically a microscopic wrecking ball.
Exactly. So in the watery environment of a living cell, having an extra reactive oxygen atom attached to a molecule's structural backbone is incredibly risky. It can cause the molecule to accidentally react with the surrounding water and just spontaneously fall apart.
But DNA doesn't have that problem.
Right, because DNA lacks this specific oxygen atom because it is deoxy. It doesn't have that vulnerability. It doesn't randomly react with water. It is chemically inert, meaning it just sits there safely.
So by simply removing one reactive wrecking ball from the structure, nature created a molecule that doesn't rust. It just sits there perfectly stable, ignoring all the chaos of the water around it.
It is the ultimate archival storage medium. And, this specific chemical stability is exactly why we occasionally see those mind-blowing news headlines about scientists extracting DNA from woolly mammoths.
Oh, like the ones frozen in the Siberian permafrost for 10,000 years.
Exactly. Or extracting readable DNA from the bones of Neanderthals who walked the Earth 40,000 years ago.
And that's because the molecule is literally engineered to survive across deep time, provided the environmental conditions don't, destroy it. If it had that extra oxygen atom, those mammoth bones wouldn't yield anything.
Not a chance. If it had that oxygen, the genetic information would have degraded into chemical dust within days or maybe weeks after the animal died. DNA's lack of oxygen allows it to persist. It's a profoundly elegant evolutionary design choice for long-term memory storage.
Okay, so we have our master blueprint. It's safe. It's missing a destructive oxygen atom, so it's ultra-stable, and it's locked in the restricted library of the nucleus.
Yep.
But this brings us to a massive practical logistical problem. The actual construction site where proteins are built, the factories, the raw materials, the machinery, is located entirely outside the library, out in the main body of the cell.
Right, an area called the cytoplasm.
So if the DNA master book cannot physically leave the restricted section of the library, but the workers outside need the instructions to build a protein, how do the instructions cross that barrier?
And this spatial problem is solved by our second main character. Enter RNA, ribonucleic acid.
The versatile worker.
Exactly. If DNA is the heavy, permanent, archival hard drive, RNA is the lightweight, temporary flash drive. RNA acts as the messenger.
Okay.
It has a structure that is very similar to DNA, but with a few critical defining differences. First, and most importantly, it is not missing that oxygen atom. RNA has the standard ribose sugar.
So RNA still has the chemical wrecking ball attached to it?
It does, which makes RNA far more reactive and structurally fragile than DNA.
Got it.
Second, while DNA is locked into that rigid double-stranded helix, RNA is typically single-stranded. Because it doesn't have a partner strand holding it tight, RNA is incredibly flexible. It can bend, loop, and fold up into complex three-dimensional shapes.
Okay, so it's bendy and fragile.
Right. And third, it uses almost the exact same four-letter alphabet as DNA.
Right.
It uses A, C, and G, but it completely replaces thymine, the T, with a different letter called uracil, or U.
Okay, I'm going to push back heavily here. We just spent all this time establishing how brilliantly designed DNA is. It's stable, it's safe, it has a backup copy literally attached to it in the double helix.
Yeah.
Why does the cell suddenly introduce this fragile, single-stranded molecule with a weird replacement letter? Why not just use DNA for everything? Why couldn't the cell just make a smaller, temporary copy of DNA, send that out of the nucleus, and build from that?
That is the logical question to ask. But to answer that, we actually have to rewind the clock much further back than the woolly mammoths.
Right.
So to go back roughly 4 billion years to the primordial dawn of life itself.
Oh, wow. Okay.
There's a widely accepted evolutionary framework called the RNA world hypothesis, and it flips our common sense completely upside down. It suggests that DNA did not come first. RNA came first.
Wait, really? RNA is the older ancestral technology.
Yeah. Long before complex cells existed, the primordial soup of the early Earth-think volcanic vents and chaotic chemical pools was likely dominated by RNA.
Why RNA?
Because RNA is single-stranded and flexible, it can fold into intricate 3D shapes. This made it the ultimate biological multi-tool. It could store genetic information in its sequence of letters, but its complex shapes also allowed it to act as a machine, physically catalyzing chemical reactions.
So it was a one-stop shop. It was the hard drive and the engine combined.
Exactly. Early life was just RNA doing absolutely everything, storing the data and doing the physical labor.
But what changed? Why do we move away from that?
The fragility was the limiting factor. Because of that extra oxygen atom we discussed, RNA constantly falls apart. In the hot, volatile environment of early Earth, relying on RNA to store your long-term genetic memory was incredibly risky.
I can see that.
It was great for rapid-fire, chaotic biology, but terrible for building a stable, complex organism that needed to pass down reliable instructions.
So I can see the evolutionary pressure building here. You need something more stable.
Right. And over millions of years, evolution stumbled upon a chemical upgrade. By removing that one reactive oxygen atom from the backbone, and by pairing two strands together to protect the letters on the inside of a double helix, a new ultra-stable molecule was born, DNA.
And DNA just took over the storage job.
Exactly. DNA essentially took over the specialized job of long-term data storage. It became the vault, freeing up the older, more versatile RNA to handle the day-to-day messenger work and the structural heavy lifting.
That reframes everything. DNA isn't the master that invented RNA. DNA is just the heavy-duty upgraded storage drive that evolved later to keep the original RNA system safe.
Spot on.
But what about the letters? Why does RNA use uracil instead of thymine? If DNA uses T, why does RNA bother using U?
It comes down to a concept we can think of as cellular economics. Manufacturing the letter thymine requires a significant amount of metabolic energy and resources from the cell. It is an expensive molecule to build.
Okay.
Uracil, chemically speaking, is essentially a cheaper, low-energy version of a thymine.
To the budget letter.
Exactly, the budget letter. Since RNA is designed to be a temporary, disposable messenger, something the cell prints out, uses for a few hours, and then breaks down, it makes absolutely no sense to spend high amounts of energy building it with expensive thymine.
That makes perfect logical sense.
Right. The cell reserves the expensive, highly stable thymine exclusively for the permanent DNA archive and uses the cheaper uracil for the disposable RNA messages.
So if you are printing a permanent wedding certificate, you use expensive, heavy stock archival paper. But if you are printing a temporary flyer for a garage sale, you use the cheapest printer paper you can find because you know it's going to end up in the recycling bin by tomorrow.
That is a perfect way to look at it. And that temporary flyer is what we call messenger RNA, or mRNA for short. And the process of making that specific flyer is the first active step in the central dogma. It is a process called transcription.
Transcription. Okay, let's go back to our restricted library analogy. We know we can't take the ancient reference book out of the room. So you take a piece of cheap scratch paper, you walk up to the pedestal, and you make a photocopy of just the one single page you need to build today's project.
Yes.
That temporary photocopy is the mRNA. But how does the photocopier actually work on a molecular level?
Well, the cell uses a complex molecular machine called RNA polymerase. You can picture RNA polymerase as the physical photocopier. Let's say you just ate a heavy meal, your blood sugar is rising, and your pancreas cells urgently need to build the protein insulin.
Okay, they need the insulin blueprint.
Right. The RNA polymerase has to locate the specific gene, the specific paragraph for insulin, somewhere in the massive DNA library.
Wait, I have to stop you there. I am stuck on the mechanics of this. You say the RNA polymerase locates the gene, but RNA polymerase is just a cluster of atoms. It doesn't have eyes, it doesn't have a tiny brain or a GPS system. How does a blind molecule navigate billions of letters to find the exact starting point for insulin?
This is where we have to discard our human intuition about how things move. We tend to imagine the inside of a cell like a calm swimming pool, where molecules swim purposefully from point A to point B.
Yeah, that's exactly what I was picturing.
The reality is that the microscopic world is governed by something called Brownian motion. It is less like a swimming pool and more like a violent, high-speed mosh pit.
Mosh pit.
Yeah. Molecules are constantly colliding with water, being thrown against each other millions of times a second by sheer thermal energy. RNA polymerase isn't looking for anything. It is tumbling and bouncing blindly along the DNA strand at blistering speeds.
So it's just pure chaos.
It is, but there's a trick. Just before the start of the insulin gene, there is a specific sequence of DNA letters called a promoter. This promoter region has a very unique physical shape and magnetic charge.
Like a highly specific landing pad.
Yes. As the RNA polymerase tumbles wildly through the mosh pit, it constantly bumps into the DNA. Most of the time, the shapes don't match and it bounces off. But when it randomly bumps into the promoter sequence, the physical shapes and magnetic charges click perfectly together, like two magnets snapping into place.
Wow.
The machine docks. No brain, no eyes, just the inevitable result of thermodynamics and physical affinity.
That is deeply unsettling, but incredibly fascinating. We are essentially running on billions of blind, lucky collisions that are chemically rigged to lock together.
Exactly.
Okay, so the photocopier has snapped onto the landing pad right at the start of the insulin gene, but the DNA is twisted shut. How does it get inside to read the letters?
Once docked, the RNA polymerase physically forces the DNA double helix to unwind and unzip, but only in a tiny localized bubble. It breaks the weak bonds holding the two sides of the ladder together, exposing the letters on the inside.
You're creaking the ladder. Doesn't that damage the master copy? We just said we can't damage it.
But it's entirely temporary. Think of a zipper on a jacket.
Yeah.
You can pull the zipper down to open a small section, but you can zip it right back up behind you.
Oh, I see.
As the RNA polymerase opens this small bubble, it reads the exposed DNA letters on one side. And as it reads, it starts stringing together free-floating RNA building blocks to build the complementary mRNA strand.
It's matching the puzzle pieces again.
Exactly. The rules of attraction take over. If the polymerase reads a C on the exposed DNA, a free-floating G naturally snaps into place. If it reads a G, a C snaps in. If it reads a T, an A snaps in.
And what if it reads an A on the DNA? Normally A pairs with T, but we are building RNA now.
And RNA doesn't use the expensive thymine. So when the polymerase encounters an A on the DNA, the cheaper uracil, the U, snaps into place on the growing mRNA chain.
So it just rockets down this unzipped bubble, reading the DNA and spitting out a trailing string of mRNA scratch paper.
It does, and it moves at an astonishing speed of roughly 40 to 50 letters every single second.
Every second.
Yeah. And the most brilliant part is the cleanup. As the RNA polymerase moves forward, the DNA immediately zips itself back up behind the machine. The two halves of the ladder snap back together, completely unharmed. The master reference book is closed, safely back on its pedestal.
And the newly minted mRNA photocopy is released. It has a copy of the insulin instructions, and it leaves the restricted library, traveling out into the bustling cytoplasm where the construction workers are waiting.
Exactly.
But I want to circle back to my earlier pushback. If this RNA photocopy is so fragile and prone to breaking down because of that extra oxygen, doesn't that mean the message might degrade before the cell can even finish building the protein? Isn't that dangerous?
It would be if the degradation were purely accidental.
Hmm?
But the cell has actually evolved to use that fragility as a highly controlled timer. The temporary nature of mRNA is a profound feature, not a bug.
How so?
Think about what happens when you cut your finger. Your cells urgently need to build clotting proteins to seal the wound. The DNA unzips, the RNA polymerase makes mRNA photocopies of the clotting instructions, and the factories start pumping out the proteins.
Right.
But once the cut is sealed, you desperately need the factories to stop. If that mRNA photocopy was as stable as DNA and lasted forever, your factories would never stop reading it.
Oh, wow.
They would endlessly churn out blood clotting proteins until your entire circulatory system solidified and you died.
Oh, so the message literally has to self-destruct.
Exactly. The cell utilizes the natural instability of RNA. There are enzymes in the cytoplasm whose sole job is to hunt down old mRNA and shred it. Depending on the specific protein, an mRNA molecule might last a few minutes or maybe a few hours before it's broken down into basic raw materials and recycled.
That's brilliant.
It guarantees that the cell only builds what it needs for the exact duration it needs it, and then the system goes quiet.
That is a masterclass in biological engineering. The fragility is the off switch. Okay, so we have completed transcription, we've copied the blueprint, the mRNA scratch paper has arrived at the construction site.
Yes.
But we are about to hit another massive logic puzzle. The instructions on the mRNA are written in an alphabet of only 4 letters: A, U, C, and G.
Right.
But the proteins we are trying to build aren't made of genetic letters. They are built out of completely different physical materials, right?
They are. Proteins are constructed from physical building blocks called amino acids. In the human body, there are 20 different standard amino acids that are strung together in various combinations to build every single protein you need.
So we have an instruction manual written in a language with only 4 letters, and it needs to command a construction crew that uses 20 different building materials. How do you translate a 4-letter alphabet into a 20-piece toolkit?
This was actually one of the greatest, most fiercely debated mathematical mysteries in biology during the mid-20th century. How does the code actually work? Let's walk through the math together, slowly, just like the scientists had to.
Okay, I'm ready. Let's do the math.
If the cellular machinery read the mRNA sequence just one letter at a time, an A might mean grab amino acid number 1, a U might mean grab amino acid number 2, and so on.
You only have 4 letters, so that only gives you the instructions for 4 amino acids. You would have 16 amino acids left over with no instructions. That doesn't work.
Right. 4 to the power of 1 is 4. So what if the machine read the letters in pairs? It reads them 2 at a time: AU, AC, AG, UU, and so forth.
Okay, well, let's do the math on that. You have 4 options for the first position and 4 options for the second. 4 times 4, that gives you 16 possible combinations.
We need to code for 20 amino acids, so we are still short.
So reading in pairs isn't enough either. It has to be groups of 3.
Let's test it. If the machine reads the letters in triplets groups of 3, you have 4 options for the first letter, 4 for the second, and 4 for the third. 4 times 4 times 4.
64 possible combinations, which is more than enough to cover all 20 amino acids.
Exactly. Nature's settled on a triplet code. The cellular machinery reads the mRNA sequence in groups of 3 letters at a time. And this 3-letter group is called a codon.
A codon.
Right. It is a literal decipherable language. For instance, if the machinery reads the codon CUU, that specific sequence translates to the amino acid leucine. If it reads GGU, that translates to the amino acid glycine.
Wait, if there are 64 possible 3-letter combinations, but we only have 20 amino acids, what happens to the other 44 combinations? Are they just blank?
They aren't blank. The genetic code is heavily redundant. Multiple different codons can translate to the exact same amino acid. This is actually one of the most brilliant defense mechanisms in biology.
How does redundancy defend the cell?
Let's use leucine as an example again. Leucine is coded by CUU, but it's also coded by CUC, CUA, and CUG. Notice a pattern.
Yeah, the first 2 letters are always CU. Only the 3rd letter changes.
Precisely. We call this the wobble position. If there is a mutation, like a random typo in the DNA, and the third letter gets accidentally swapped from a U to an A, the code changes from CU to CUA.
Oh, I see.
But because of the redundancy, the machinery still grabs leucine. The typo happened, but the final physical protein remains perfectly intact. It is a built-in molecular autocorrect.
That is staggering. The system anticipates its own errors and absorbs them. But if this mRNA is just a massive continuous string of thousands of letters, how does a machine know where to start reading? If it starts one letter too early or one letter too late, all the three-letter groups shift and the entire protein will be ruined.
You've just described a frame shift mutation, which is incredibly destructive. To prevent that, the code contains specific punctuation marks. The three-letter sequence, AUG, is universally known as the start codon.
Like a giant capital letter at the beginning of a sentence.
Yes, it tells the machinery, do not read anything before this. Begin counting your triplets exactly here.
So AUG sets the frame.
And conversely, there are three specific combinations, UAA, UAG, and UGA, that do not code for any amino acids at all. They are the stop codons.
The period at the end of the sentence.
Exactly. When the machinery hits one of those, it knows the protein is finished, and it releases it.
Okay, so we have the start code, we have the ingredient list, and we have the stop code. The translation manual is complete. But knowing the language doesn't physically build the structure. I mean, an architect's blueprint doesn't magically stack the steel beams. You need heavy machinery. You need cranes, workers, and delivery trucks.
Which brings us to the second and perhaps most physically complex phase of the central dogma, translation. We're going to translate the abstract RNA message into physical matter. And to accomplish this, two entirely new types of RNA enter this story.
Okay.
Up until now, we've only talked about messenger RNA, or mRNA. Now, we need to introduce ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA.
Let's break down the heavy machinery first, the factory itself. Ribosomal RNA.
Ribosomal RNA folds up and combines with dozens of proteins to form a massive, complex molecular structure called a ribosome. You can think of the ribosome as the central cellular workshop. But honestly, a more accurate modern analogy is a molecular 3D printer.
Oh, I love the 3D printer analogy.
Yeah, the ribosome is the heavy machine that physically clamps onto the mRNA photocopy, reads the three-letter codons, and prints the physical protein chain.
So the mRNA thread feeds into this ribosome printer like a ticker tape. The printer reads the first codon, let's say it's that start codon, AUG. The printer knows it needs the specific amino acid assigned to AUG. But where does the amino acid come from? The printer can't just manifest matter out of thin air.
That is where the third type of RNA arrives, transfer RNA or tRNA.
Right.
If the ribosome is the 3D printer, the tRNAs are the delivery trucks. They're small, intricately folded RNA molecules that constantly swarm and float through the cytoplasm.
Okay, but trucks.
Right, and a tRNA has two very important ends. One end is a three-letter sequence called an anticodon. This anticodon is physically shaped to perfectly match and bind with the mRNA codon.
And the other end?
The other end of the tRNA truck carries the physical cargo, the specific amino acid that corresponds to that code.
So they act as literal physical adapters. One end plugs into the information on the mRNA, and the other end delivers the physical matter, the amino acid.
It is the ultimate bridge between information and physical reality.
I want to visualize this assembly line. How does this fast-paced ballet actually work inside the 3D printer?
Well, inside the ribosome, there are three distinct docking stations arranged side by side where these tRNA delivery trucks pull in. They're called the A slot, the P slot, and the E slot. Let's walk through the conveyor belt.
A, P, and E. Okay, I'm picturing 3 parking spaces inside the printer.
The mRNA ticker tape is threaded through the bottom. The first three-letter codon is exposed in the first slot, the A slot, which stands for arrival. A swarm of different tRNA trucks is constantly bouncing in and out of that slot due to Brownian motion.
Right, the mosh pit again.
Most of them bounce right back out because their anticodon doesn't match the mRNA. But eventually, the truck with the perfect matching anticodon bumps in, and it chemically clicks firmly into place.
The first truck has arrived and parked in the A slot.
Now, the ribosome machine physically shifts the mRNA ticker tape down by exactly 3 letters. When the tape moves, it drags that first parked tRNA truck with it, pulling it into the middle parking space, the P slot, which stands for processing.
Okay, so the first truck has moved to the middle. That means the A slot is now empty again, and a new 3-letter codon is exposed there.
Exactly. A second tRNA delivery truck bounces in. Its anticodon matches the new codon, and it docks in the A slot. So now you have 2 trucks parked side by side, the one in the P slot and the new one in the A slot.
Got it.
Both are holding their specific amino acids up in the air. Here's the magic moment. The ribosome acts as a welder. It breaks the bond holding the amino acid to the first truck and chemically welds that amino acid onto the cargo held by the second truck.
It's linking the physical chain. So the first amino acid is now attached to the second one.
Right, it creates a strong peptide bond.
No.
Now, the second truck in the A slot is holding a chain of 2 amino acids. The ribosome shifts the ticker tape down by 3 letters again. The first truck, which has now dropped off its cargo and is completely empty, gets pushed into the final parking space, the E slot.
E for exit.
Exactly. It detaches from the mRNA and floats back out into the cellular fluid, empty, ready to go pick up another amino acid. Meanwhile, the second truck, carrying the growing chain, has been pulled into the middle P slot. The A slot is empty for a third truck.
And the cycle just repeats?
Yep. Arrive, process, exit. Arrive, process, exit. Over and over, sometimes thousands of times, welding amino acid after amino acid into a massive, complex chain.
And this rapid-fire assembly line just keeps chugging along until the A slot exposes one of those stop codons, like UAA.
Exactly. When a stop codon is exposed, there is no tRNA truck that matches it. Instead, a protein called a release factor docks there. It basically tells the 3D printer, we are done. The printer releases the long chain of amino acids. Driven by the magnetic charges of the amino acids themselves, the chain instantly folds up into a highly specific 3D shape, and boom, you have a fully functional protein ready to keep you alive.
It is breathtaking to imagine this happening inside my arm right now, but I have to stop us again. I see a glaring logical flaw in this delivery system, a massive vulnerability.
Okay, let's hear the counter-argument.
You said that the empty tRNA delivery truck gets ejected from the E slot and it floats away into the cellular soup to go pick up a new amino acid so it can be used again.
Yes.
But tRNA is just a dumb piece of folded RNA. It doesn't have a brain. How does the empty truck know to pick up the correct amino acid? If a tRNA is genetically coded to carry leucine, but it accidentally bumps into a glycine molecule out in the soup and picks that up instead, the whole delivery system is compromised.
That's true.
Right, because the 3D printer would grab that truck, read the leucine code, but physically weld a glycine into the protein. The code would be printing absolute garbage.
You have just zeroed in on what is arguably the most vital, unsung quality control mechanism in all of biology. You are completely right. The ribosome 3D printer is blind to the cargo.
It doesn't check.
No, it only checks the 3-letter anticodon on the bottom of the truck. It never checks what amino acid is actually strapped to the top. The fidelity of the entire central dogma, the reason you are alive, relies entirely on making sure the correct amino acid gets loaded onto the correct tRNA truck out in the cytoplasm before it ever reaches the ribosome.
So how does the cell prevent a massive mixing of the cargo?
Through a rigorous verification system managed by 20 highly specific inspector enzymes out in the cytoplasm. There is one distinct inspector enzyme for every single one of the 20 amino acids.
Let's avoid the textbook names here. How do these inspectors actually work? Because I keep picturing little sentient workers with clipboards, which we know isn't true. They are just molecules.
Right. Think of the inspector enzyme not as a worker, but as the most complex, restrictive, 3-dimensional lock you can imagine. This enzyme has 2 very specific binding pockets.
Okay.
One pocket is shaped to perfectly accommodate only one specific amino acid, let's say leucine. The other pocket is shaped to perfectly accommodate only the specific tRNA truck that possesses the anticodon for leucine.
It's a dual verification lock.
Exactly. Due to Brownian motion, random amino acids and random tRNAs are constantly bouncing into the inspector. But unless both the correct amino acid and the correct tRNA dock into their respective pockets simultaneously, nothing happens.
And when they do match?
When the perfect match occurs, the inspector enzyme acts as a press, physically welding the cargo onto the truck.
So it guarantees the match through pure physical geometry.
Yes. And making this specific weld requires a massive investment of cellular energy. The inspector uses a molecule called ATP, which is the universal energy currency of the cell, and basically burns it to permanently lock the amino acid onto the tRNA.
That sounds expensive for the cell.
It is. But by investing this high amount of energy exclusively at the quality control stage, the cell mathematically guarantees that the genetic code translates perfectly into physical matter. If this locking step failed, biology as we know it would collapse into chaos.
I want to take a breath and just summarize this unbelievable sequence.
Sure, go for it.
Okay. The DNA, our master blueprint, sits safely in the restricted library, protected from rust because it lacks an oxygen atom. When a protein is needed, RNA polymerase bumps into the promoter, unzips a tiny bubble, and builds a temporary disposable mRNA photocopy.
Yep.
That fragile photocopy travels out to the cytoplasm where the ribosome 3D printer clamps onto it. Meanwhile, those physical lock and key inspector enzymes are out in the fluid, burning heavy energy to load the correct amino acid cargo onto the tRNA delivery truck.
Exactly.
The trucks arrive at the printer, locking into the A, P, and E slots, matching their 3-letter codes to drop off their cargo. The amino acids are welded into a chain, the chain hits a stop codon, it folds up, and you have a protein.
You have just elegantly described the fundamental mechanism that allows life to exist, to adapt, and to build itself from raw chemistry.
So why does all of this microscopic detail matter to the person listening right now? We promised at the beginning that we were going to connect this abstract biology to the real world. Why is understanding the central dogma the key to the future?
Because once you truly understand the central dogma, you possess the key to modern medicine.
Oh, really?
Yeah. For centuries, medicine has been reactive. We try to kill invaders or cut out tumors. But by understanding how the cell's 3D printer works, we don't have to rely on traditional brute force methods. We can simply hand the body the instructions to heal or protect itself.
Give me an example.
The most famous world-changing application of this knowledge is the mRNA vaccine technology, which actually won the Nobel Prize in physiology or medicine.
Oh, this is the technology that pulled us out of the COVID-19 pandemic.
Let's compare it to the old way of doing things. For decades, traditional vaccines required a massive physical infrastructure. Scientists would have to grow billions of actual viruses in giant vats of chemicals or inside millions of chicken eggs.
Chicken eggs?
Yeah, millions of them. They would then kill or severely weaken the virus and inject that physical dead virus into your arm so your immune system could get a look at the shape of it and learn how to fight it.
Growing vats of dangerous pathogens sounds slow, expensive, and frankly, logistically terrifying.
It is incredibly slow. But once researchers mastered the central dogma, they asked a revolutionary question. Why are we spending months growing physical proteins in vats? Human cells are already packed with billions of molecular 3D printers that run 247.
The ribosomes.
Right. Instead of injecting the physical virus, why don't we just inject a temporary photocopy of the instructions and let the human body's own ribosomes build the training target?
That perfectly maps onto everything we just discussed. You just send a text message to the factory.
Exactly. The COVID-19 virus, SARS-CoV-2, has a shell covered in distinct little spikes, which we call the spike protein. Scientists sequenced the virus's genome and found the exact paragraph of letters, the gene that codes for that specific spike.
And they ignored the rest of it.
They didn't need the rest of the virus. They just synthesized a pure mRNA photocopy of the spike instructions in a lab.
They printed a fake blueprint.
Yes. They wrapped this highly fragile synthetic mRNA in a tiny protective bubble of fat called a lipid nanoparticle. And that is what gets injected into your shoulder.
And when that fat bubble enters my cell, it releases the synthetic mRNA into the cytoplasm. And as we learned, the ribosomes, the 3D printers, don't have brains. They don't check a security badge to see if the mRNA came from the nucleus or from a syringe. They just clamp on and start reading.
The tRNA delivery trucks start bringing in the amino acids matching the codons. The A, P, and E slots fire away. And what does your ribosome print? It prints thousands of viral spike proteins.
But importantly, just the harmless spike. Not the dangerous internal machinery of the virus, just an empty piece of the shell.
Right. It's like your cell is manufacturing a harmless training dummy. Your cell displays these printed spikes on its surface. Your immune system patrols by, spots this totally foreign, strange-looking protein, and goes into high alert.
It learns the shape.
It learns the exact physical shape of the spike, builds specialized antibody weapons to destroy it, and creates a permanent memory cell. If the real virus ever infects you months later, your immune system already has the weapon fully calibrated and destroys the virus before you even feel a fever.
But wait, a common fear people had was about the mRNA staying in their body. What happens to the synthetic mRNA after the printer reads it? Does it alter your DNA? Does it just sit there printing spikes forever?
Think back to the chemical structure of RNA. Why did RNA evolve into DNA?
Because RNA has that extra oxygen atom. It's chemically fragile. It's designed to be temporary.
Precisely. mRNA is inherently unstable. It is a disposable message. Once your ribosomes have read the vaccine's mRNA a few times, the natural shredder enzymes in your cytoplasm recognize it and tear it apart.
It's gone.
Completely. The synthetic mRNA is completely degraded into basic cellular raw materials within a few days. It never even physically enters the protected library at the nucleus. It cannot touch your DNA. It simply delivers the message, prints the training dummies, and dissolves back into the soup.
That is the most elegant medical intervention imaginable. We quite literally hacked the central dogma. We learned the ancient triplet language of the cell, and we used it to upload a defensive blueprint.
An infectious disease is really just the opening chapter. Now that we have the delivery system mapped out, the possibilities for oncology are staggering.
Cancer treatment.
Yeah. We can biopsy a patient's unique cancer tumor, sequence its DNA, and find the mutated proteins that are specific to their cancer alone. We can synthesize an mRNA blueprint of those cancer proteins, inject it back into the patient, and have their own body print the targets.
Wow.
We can train the immune system to hunt down and eradicate cancer cells with microscopic precision.
Personalized, custom-built cancer vaccines, that is the holy grail of medicine. And it is entirely possible because we finally understand the flow of information from DNA to RNA to protein.
It is the ultimate validation of basic foundational research. By understanding the mechanical physics of life, we gain the power to protect it.
As we bring this deep dive to a close, I want to recap the territory we've covered today.
It's been quite a journey.
It really has. We started with the foundation. DNA is the master blueprint, locked safely in the restricted library of the nucleus, stable and unchanging because it lacks a destructive oxygen atom.
Right.
We explored the primordial RNA world and how RNA evolved to become the disposable, flexible messenger. We watched RNA polymerase navigate the chaotic mosh pit to transcribe the temporary mRNA photocopy.
The scratch paper.
Exactly. We decoded the triplet math of the 64 codons, marveling at the redundancy that protects us from mutations. We explored the ribosome 3D printer and watched the tRNA delivery trucks drop off their amino acid cargo, perfectly verified by the heavy energy locks of the inspector enzymes.
The sheer mechanical coordination required for a single breath or a single heartbeat is awe-inspiring.
And the most profound takeaway is that this isn't just an abstract theory we are discussing. Right this very second, as you sit here listening, millions of RNA polymerases are violently zipping along your DNA, opening tiny bubbles and spitting out fragile mRNA photocopies.
It's happening constantly.
Billions of tRNA delivery trucks are physically locking into the A, P, and E slots inside your cells, welding amino acids together. You are a walking, breathing masterpiece of constant biological engineering, running entirely on a 4-letter code.
It truly is a marvel. But before we sign off, I actually want to leave you with a final provocative thought to ponder. Something that builds on everything we've just learned, but introduces a whole new layer of biological mystery.
I always appreciate a good twist ending. What do you have for us?
We established very early on that every single cell in your body contains the exact same DNA library. The DNA blueprint sitting in the nucleus of your brain cells is completely mathematically identical to the DNA blueprint sitting in your skin cells, or your liver cells, or your heart cells.
Right. It's the same master reference book copied into every cell when I was an embryo.
So here's the paradox. If the blueprints are exactly the same, how does a heart cell know to build the proteins that make it beat, while a skin cell builds the flat structural proteins to form a barrier? If they have the exact same instructions, why don't the 3D printers print the exact same protein?
Wait, if the instructions are identical, the output should be identical. Why aren't we just a giant uniform blob of the same cells?
The answer lies in a fascinating frontier field called epigenetics. The prefix epi means above or on top of genetics. It turns out your DNA sequence is fixed, yes. The sequence of A, T, C, and G doesn't change. But how that blueprint is accessed can be heavily modified. Your environment, your diet, your exercise habits, and even the trauma and stress you experience can cause your cells to place chemical padlocks directly onto the DNA itself.
Chemical padlocks?
Yes.
No.
Small molecules called methyl groups can chemically attach to the DNA ladder, physically blocking the RNA polymerase from docking at the promoter. In a heart cell, the chapters of the book that contain the instructions for building skin proteins are heavily padlocked.
Oh.
They are physically silenced. The photocopier literally bounces off them and skips over them. In a skin cell, the heart genes are padlocked shut.
So the library is identical in every cell, but different rooms are barricaded depending on what kind of cell you need to be?
Exactly. And because these padlocks are highly sensitive and influenced by your environment, it means that your lifestyle choices can actually change which pages of your genetic library get read and which ones get silenced.
That's incredible.
You are not purely at the mercy of your fixed DNA sequence. The way you interact with the world actively influences the expression of that DNA throughout your entire life, constantly adding and removing padlocks.
Your blueprint is fixed, but the way the cellular architect reads the filing cabinet can actively change based on how you live. We started today looking at a skyscraper, marveling at the glass and the steel, and wondering about the hidden filing cabinet of blueprints.
I did.
We've opened the cabinet, we've read the ancient four-letter code, and we've watched the microscopic cranes and delivery trucks weld the building together. But to know that you can actually walk into that architect's office and place a padlock on certain drawers just by the way you interact with your environment, that changes our entire understanding of biology.
It does.
It means the skyscraper of the human body is never truly finished. It is always rebuilding, always adapting, reading the ancient blueprints in entirely new ways.
It is the ultimate dynamic system, adapting to the chaos of the world in real time.
Thank you for joining us on this incredible microscopic journey into the code of life. Until next time, keep marveling at the skyscrapers around you, but never forget the absolute magic happening right now inside the filing cabinet.